Okay, good afternoon, everyone. So we were looking at the structure of the nephron, or rather the structure of the kidneys, in terms of how the longitudinal section looks like. And if you remember, we had just looked at these 2 or 3 structures in terms of cortex and medulla, 2 separate divisions, cortex being the outermost and the medulla being the inner one. And within the renal medulla, there was this striped tissue arranged in a conical pyramids, and the tip of that pyramid was called as papilla, which is finally projecting into the pelvis of the kidney. So if you don't remember, here is the diagram. These pyramid-like structures, they are the like conical pyramids that I'm talking about in terms of striped tissues. And the apex, that is the tip of this tissue, is called as renal papilla. And the finally, all of these renal papilla that opens into the pelvis, which is one of the cavity, finally extending outward as ureter. So that was the pyramid. And then the last point that we had seen was the kidney is composed of many nephrons, right? So now we'll see how the structure of the nephron and even the function of the nephron in the later slides. So this basic part you already know that it's the structural and functional unit of the kidney. Now what does it mean by this statement? Is like obviously forming the building block of the kidney. So that's the structural unit of the kidney, and the major function of the kidney is done by the nephron. So that's why the functional unit also of the kidney. So it consists of something which is called as glomerulus. I assume that you haven't heard these terminologies before in the previous classes, like class 9 or so. So there is something called as glomerulus, which is a network of capillary and a network of blood capillaries, I must say, and it is associated tubule through which the glomerular filtrate passes before it emerges as urine. So let me go back to the diagram here. So this is the diagram of the nephron that you can see here. This particular part, which is shown as a capillary network within the this C-shaped structure, that is called as glomerulus. You can see probably on the top. I guess I will have to put it down. Yeah. Now you should be able to see. So you can see this name glomerulus, which is the capillary network. So there is a main artery which brings blood to the kidney. That will be distributed into several of its branches, making final afferent arteriole. And this afferent arteriole individually will bring the blood to different different nephrons. So one of them, let's say we are talking of this afferent arteriole, bringing the blood to this capillary network, and there will be efferent. Afferent, A stands for arriving, so blood is incoming to this capillary network. Efferent means exiting or outgoing, so that one the blood is taking away from this. So what happens is, due to the difference in the diameter of the afferent arteriole with respect to the efferent arteriole, you can see this is relatively smaller diameter, this is relatively bigger diameter, wide and narrow, right? So when the blood passes through this and then there is a smaller diameter, it increases the pressure in this capillary network, that is the glomerulus, and because of which the blood is kind of squeezed out. Not the blood as such, but the plasma from the blood is squeezed out, and that is received by the C-shaped body called as Bowman's capsule, and then it goes out. So this particular first filtrate or first squeezed out plasma from the glomerulus is called as glomerular filtrate. So that is what is being, where is it? Sorry, it's ahead. So that is what this slide is saying. So this nephron consists of a glomerulus, that is the capillary network, and its associated tubule. That means whatever tubes it is, glomerulus is associated with, through which the glomerular filtrate passes. That means whatever blood is squeezed out in terms of plasma, that is water and nutrients, that will be passing out before it emerges as the urine. So at the later stage of nephron, This is the same filtrate that will emerge or that will be excreted as urine. Nephrons are microscopic structures composed of renal corpuscle and renal tubule. So nephron has two main parts. What we commonly know as renal corpuscle. Corpuscle means like a capsule or a bulged body. So this is like a renal capsule or renal corpuscle, and the rest of this tubular structure that you see in blue, that is the renal tubule. So the nephron is composed of renal corpuscle and renal tubule. The word nephron is actually derived from the Greek word called nephros. It means kidneys, which ultimately means to filter out. So that is the meaning of this word nephron. And there are like more than millions of nephrons within each kidney. So relatively, we have quite a few millions within both the kidneys. So that way, these nephrons, they will function to filter out the urine from the blood. So the average size of the mammalian nephron is 35 to 55 millimeter long. That is 3.5 to 5.5 centimeter long, which is like still relatively very small. And this we are talking of the whole nephron. So at one of the end, the tube is closed, folded and expanded into a double-walled cup-like structure called as Bowman's capsule, or it is also called as renal corpuscular capsule. So again, I'll go to the diagram here. So if you look at this diagram, the renal tubule is expanded, folded and kind of cup-like structure that is with double layers. Double layers as in there is inner layer, inner concave shape and the outer concave shape. And you need to imagine this Bowman's capsule as a three-dimensional cup, not the C shape. Yeah. So like everyone is very much aware of how does a cup look like, right? So that is like a three-dimensional body with hollow cavity, and within which there are this glomerular capillary network. But while the diagram is being shown, it is always shown as a C-shaped body because it's like a two-dimensional representation. So that is the Bowman's capsule. So at one end, the tube is closed. That means there is no opening on that side. It is folded and it's expanded into a double-walled cup-like structure called Bowman's capsule. It is also known as renal corpuscular capsule. So it has just different names, nothing else, which encloses the cluster of microscopic blood vessels called as glomerulus. So there is a big network of capillaries or blood vessel cluster, that is what we call as glomerulus. This capsule and the glomerulus together, that consists of renal corpuscle, which is the first part of the nephron. So nephron is divided into two, renal corpuscle and renal tubule, right? Renal corpuscle consists of glomerulus and Bowman's capsule. So that's how the distinction is done between the structure. So again, in this case, whole of this thing is the renal capsule or corpuscle, and this blue part, a tube-like structure, that is renal tubule. Within this renal corpuscle, this capillary network is specifically called as glomerulus, and this cup-like object or structure is called as Bowman's capsule. So that is the distinction. Now, if you look at the real image, real as in this is not a real image, but this is quite a good representation of how it looks like in reality. So in this, if you specifically see that this is the capillary network. So if you look here closely, you can see these red blood vessels. It's coming down. One of it is branching out from here, and it is entering into the glomerulus network, right? And again, there is the other exiting one, which is taking the blood away from the glomerulus. So whatever is filtered is going to this very finely represented Bowman's capsule. You can see this kind of faint or light orange, or I don't know, whatever that color is, skin color. That Bowman capsule will receive the filtrate from the glomerulus, and it will pass down through this renal tubule, and finally it will go and enter into this main collecting duct. So we'll see all these structures. As of now, what you need to remember is renal corpuscle consists of glomerulus and the Bowman's capsule, that is the C cup-shaped object. The renal corpuscle consists of glomerulus, which is surrounded by the Bowman's capsule, that we know already now. The glomerulus arises from the afferent arteriole. So the start of the glomerulus is actually from the afferent arteriole, that is A stands for arriving, that is incoming arteriole. So one which are bringing the blood to the nephron, and then empties into the efferent arteriole, that means it is the exiting one or the outgoing one. So smaller diameter of an efferent arteriole helps to maintain the high blood pressure in the glomerulus. Now, why it has to maintain high blood pressure? The reason being because it has to filter out. It's the simpler analogy that when you are probably watering the garden with a pipe, and then you hold the tip of the pipe, the water travels further distance because the pressure is being built up. Something similar happens here, but there is no open end. So obviously the water as in plasma from the blood will filter out from this capillary network, and it will be received by the Bowman's capsule. This Bowman's capsule is divided into three main layers, what we know as outer parietal layer, middle basement layer, and the inner visceral layer. Visceral layer usually refers to the cavity layers. So outer parietal layer is made up of the epithelial cells with minute pores and of diameter 12 nanometer. So the outermost layer, that is the parietal layer, is made up of the epithelial cell, which is regular type of tissue at most of the lining with minute pores. Yes.
So, like this the third point said, smaller diameter helps to maintain high blood pressure, right?
Right.
Uh, so why does it only specifically talk about the efferent arteriole? Why not the afferent one?
Because that's what we see here. Afferent has the wider diameter, efferent has the narrower diameter. So in comparison, if you see, when the blood is coming from a diameter which is bigger to the diameter which is smaller,
The
obviously
pressure getting
it
filled.
will increase. Right? That's what it is talking about. Yeah, so that was the outer parietal layer, which is outermost layer, and it has certain pores of this 12 nanometer size. Again, this you need not remember, but you just need to remember that it is porous layer. Then the middle basement membrane, it is just selectively permeable. Selectively permeable, if you remember, it will allow the specific molecules only to pass through it. That is what is selectively permeable membrane, and that is the middle basement membrane. And the third, that is the inner visceral layer. It consists of large nucleated cells called podocytes, and they bear a finger-like projection called podocell. Now, this is obviously whatever is incoming. Again, I'll go back to the diagram. So, whatever is incoming or whatever is filtered out from this glomerular network will be received by this inner layer of the podocytes, which is the right next to the glomerulus. Then they will selectively allow some molecules to pass through the middle basement membrane, and then whatever is required by the body, it will be squeezed out through the pores which are present on this outermost layer, that is the outer parietal layer. So the inner visceral layer will capture everything that is coming out from the glomerulus. Middle basement membrane will allow it to pass through it, and the outer parietal layer will finally either let go of them out of the nephron structure, or it will continue to carry it in the nephron itself. So that way, these three layers, they are very, very important from the Bowman's capsule. So these are just structures within the renal capsule or renal corpuscle, right? Now we will look at the structures within the renal tubule, that is this tubular part. As of now, does anyone have any question so far? Okay. So then the renal tubule is a long convoluted structure. Convoluted, it literally means folding, right? Like our brain has a convoluted layer, that means it has a folded layer. So whatever, if you look at the diagram again, so these foldings, they are called as convolutions. Even if you look at the kind of actual representation, these foldings, these are called as convolutions because it's too much of the folding, either in terms of the starting part of the nephron or even the ending part of the nephron. It's kind of folded, and that is what is convoluted. So the renal tubule is long convoluted structure that emerges from the glomerulus, that is, it's coming from the renal corpuscle, and can be divided into three main parts. That is proximal convoluted tubule, Henle's loop or loop of Henle, and the distal convoluted tubule. These are the three parts. So let me show you these parts here in the representation. So the first part immediately after the renal corpuscle is the proximal convoluted tubule, PCT it is short form as. And then there is a U-shaped structure, it's called as loop of Henle. And the last part, it's distal convoluted tubule. Distal convoluted tubule, that is the last part of this, out of these three. So within the first part, that is proximal convoluted tubule, it is called so because first of all, it is proximity to the glomerulus. That's why it is called as proximal. It is convoluted, so proximal convoluted tubular structure. And it stays in the renal cortex. Now, what does it mean it stays in the renal cortex? If you look here, this part between the blood vessel is the renal cortex. And this part which is striped is the renal medulla. Again, if you don't remember, we had seen this part here. This outermost layer was the cortex, and the inner layers, these were the renal medulla. So this PCT, that is proximal convoluted tubule, it stays within the cortex. The next part, that is loop of Henle, is actually within the medulla, because you can see here this striated part or this striped muscle, it has this loop of Henle, like this U-shaped thing, which is shown here like a perfect U shape, but that is again a diagram. So the second part is the loop of Henle, or it is also called as nephritic loop, because it forms a loop with ascending and descending loop, and that goes through the renal medulla. First part is in cortex, proximal convoluted tubule. Second part is in renal medulla. And the third part is again in cortex, that is distal convoluted tubule. And again, why it is called as distal convoluted tubule is because it is far away from the glomerulus or far from the renal corpuscle, that is the distal nature. So distal, and again it is folded, so convoluted tubule. So this way, the nephron is divided into two main parts, renal corpuscle, renal tubule. Corpuscle is divided into glomerulus plus Bowman's capsule, and the renal tubule is divided into PCT, loop of Henle, and DCT. So these are the three main parts. And like I said, whole of this structure, upper one, that is glomerulus, PCT, and DCT, is within the cortex, and loop of Henle is within the medulla portion of the kidney. So that's just the basic information of whole of this structural aspect. Does anyone have any question here?
So, just asking, other than the structure of the nephrons, what else do we have in the chapter?
Function.
Like, yeah, of course. Structure, function, that's all, right?
Then,
That's all.
yeah.
Okay, okay. Nice, nice.
Okay, so these are the three parts, and like the last point just says that the Bowman's capsule will extend to form PCT. PCT continues to the loop of Henle. Loop of Henle ascends into DCT, and which ultimately goes into the collecting duct. So that's the sequence, how it goes from one place to the other. So from glomerulus to Bowman's capsule to PCT to loop of Henle, then to DCT, and then finally the collecting duct. So this big pipeline or tube-like structure, that is the collecting duct, because it's actually collecting from all the different nephrons. So if you see this diagram, it has many openings or branches. You can see here, here, here. All of these different branches, they will be collecting from different nephrons. So within one that conical pyramid itself of the kidney, there will be many nephrons. And then there are many such conical pyramids. So that means ultimately even thousands or even more than that within one small area of the nephron. And imagine all these structures in the three-dimensional, so that this will be more easier to understand. Okay, so then the main function of these tubules, that is the renal tubule, is reabsorption, and the process can either be through the active transport or passive transport. Now you know what is active and what is passive, so I need not speak about that here. But there are two main functions, out of which one being the major one being reabsorption. That means whatever is gone out from the blood has to be reabsorbed back, because that might be required by the body cells. So that is the reabsorption function of the tubule. And the second one being, in addition, secretions by the tubules. So that is the second function of secretion. And this part we'll see in the final function of the nephron as well. So that secretion by tubules help in the urine formation without affecting the electrolyte balance of the body. So whenever this there is quite a lot of excess of, let's say, glucose or what we say, salts and so on. So in all of these cases, or higher amount of electrolytes or higher amount of particular compound, if it is too much of it within the blood, that has to be secreted out in the urine, and that particular function is known as secretion of the tubules. And this is specifically required to maintain the electrolyte balance or the homeostasis within the body. So whatever water is gone out can be reabsorbed, or if it is in too much of excess, it will be secreted out into the urine. Then the first, coming back to the first part of the nephron, that is proximal convoluted tubule, and what exactly happens there. So the blood is brought to the kidney by renal artery, and then it is filtered by the glomerulus, and what we receive is glomerular filtrate. That is the first filtrate, which is not urine at all, which is just the filtrate of the blood. And then it is passed down to the proximal convoluted tube. Now within the PCT, which has highest concentration of all the nutrients or all the electrolytes, vitamins and whatsoever, right? So maximum reabsorption takes place in the PCT itself. That is the proximal part of the renal tube. Maximum reabsorption means whatever is gone or whatever is filtered from the blood to the Bowman's capsule has to be taken back, because body might need them at times. Like for simplest example, if there is lot of glucose in the blood and that blood or that plasma with glucose is being separated into a nephron, obviously glucose can be utilized by any type of cells. So all of that glucose has to be reabsorbed from the PCT, and that is the first step. So PCT is the region of renal tubule where the reabsorption of essential substances like glucose, proteins does not mean big protein, these are very smaller proteins, amino acids and major portion of the electrolytes and water takes place. So all of these molecules, they are being reabsorbed into the PCT. And the surface area for reabsorption is facilitated by the lining of the simple cuboidal epithelium within this PCT region. And then the reabsorption takes place at the expense of energy because it's an active process. And that will be quite obvious as well, because already in the blood it will have higher concentration. And from glomerular filtrate to absorb it back into the blood, that will be against the concentration gradient, and that is why it is an active active process. So that is the first part of the PCT. Along with this, PCT selectively secretes ions. Depends on which ions they are and how much amount is present within the blood. But this PCT will secrete these ions like hydrogen, ammonia, potassium into this glomerular filtrate and will absorb the bicarbonate from it. And thus PCT will maintain the electrolyte and acid-base balance of the body fluid. That means based on how much hydrogen is going out or hydrogen is being retained, or similarly for bicarbonate being removed or retained, the pH and the acid-base balance of this body fluid can be maintained by this PCT itself. Again, any question here? Anyone? Everyone has gone to the sleep. I hope you are able to follow it even if you are feeling sleepy. Because it's actually very theoretical here, that part. I guess we will only take the Henle's loop and then we will stop. The remaining part we can cover it in the next class. So with respect to Henle's loop, it has a descending and ascending loop. Now what does it mean by descending and ascending loop? Usually by virtue of saying it, we say first one is ascending and second one is descending. But in case of Henle's loop, if I show you the diagram, the one which goes down, that is descending loop. And then the one which comes up again, that is the ascending loop. So that way the first one is descending loop, then the U shape, and then the ascending loop. So even here, the glomerulus, then PCT, which is most convoluted part shown here. And this one is descending loop. And then it again starts rising up, which is ascending loop. And then it enters into the collecting duct. One most important parameter that we must make sure that we know it is the diameter of the Henle's loop is very thin compared to again these other parts, that is DCT and PCT. The diameter of Henle's loop is very thin, and there are reasons for that, why we will see that. But in representation you can see that it is not shown like that, but in reality Henle's loop has difference of diameter compared to PCT and DCT. So that is the Henle's loop. Being parts of the same loop, both the descending and ascending loop, they show different permeability.
So sorry to interrupt.
Yeah.
You said that Henle's loop has a smaller diameter.
Yes.
Oh, okay.
A part of it.
Okay.
So this both ascending and descending loop, they have different permeability, and why and how, that is what is the next point. So the descending loop, that is the first part of the Henle's loop, is permeable to water and it is not permeable to the electrolytes or whatever the minerals are present. So it will only allow water to pass out, and it will not allow the electrolyte to pass out through it. That is the descending limb. That is the first part of the loop. While the ascending limb, that is the second part of the Henle's loop, is only permeable to electrolytes and it is impermeable to water. So what happens is when the, let me go back to the diagram. So let's say there is a glomerular filtrate which is received by the Bowman's capsule. It will be traveling through the PCT where maximum reabsorption of the nutrients and water will happen. The remaining filtrate or remaining remnants of that filtrate will come down in the descending limb. In the descending limb, most of the water will be absorbed and the electrolytes cannot pass through this descending limb. So that is first part. Then when it goes further, the water cannot be absorbed at all, but the electrolytes can be absorbed by the ascending limb. So that way, these two limbs of the Henle's loop, they have different function. First, only water is permeable, electrolytes or minerals are not. And secondly, water is not permeable, but the electrolytes are permeable. So in the first case, when the water is permeable, that means water is moving out. So the solution is getting more concentrated. And in the second case, because it is what we say, water is intact and the electrolytes are getting absorbed, then it remains as a dilute solution. So that was the last point. So the surface area, so the electrolytes, they get reabsorbed at the ascending loop of Henle and the filtrates gets diluted as it moves towards the ascending loop. That is the second part. So first it will remain concentrated because water is going out, electrolyte is as it is. So it's less amount of water, more amount of electrolyte. So it is concentrated, and as it moves further in the ascending limb, it will take out most of the electrolyte as well. So the solution is becoming dilute. And the reabsorption is very limited in this segment. But in the Henle's loop, reabsorption itself is a very limited process. Maximum reabsorption has already happened in the PCT part of it. Again, any question, anyone?
This is under the heading of reabsorption. No.
That we will see further. So let me show you that part as well. So when we talk of the production of urine, there we will talk of these three things specifically: ultrafiltration, reabsorption, and tubular secretion, which we have seen in a bit to some extent, but that's where the reabsorption comes into picture. But in general, we are looking at the structural ability of a particular nephron part, and based on that, what are the functions of that particular part. That's what we are looking at. When we look at the urine production mechanism, there we will talk of which part and what mechanism and what exactly is getting filtered out and so on. So that
Just one thing. In my bio textbook, this diagram that you showed of the nephron structure with Bowman's capsule and loop of Henle, it has this note given, highly diagrammatic. What does that mean?
highly diagrammatic means it's a representation. It is not an actual representation. It has been modified to represent it in the form of a diagram.
Oh.
That's what it means by highly
Basically, it's been flattened, like 3D rotated.
Exactly. But flattened and also simplified. Okay. If anyone has any questions, I can take it. Otherwise, we'll stop here for today.